Morning, all of you. I hope everybody is fine. In today's video, we're going to talk all about auxin. First, let's see what auxin looks like. This is the molecular structure of auxin. Don't worry, you don't have to remember this structure. I'm just introducing. But you do have to know its scientific name. Auxin is called indole acetic acid, short form IAA. And it is made from the precursor amino acid called tryptophan. So we move on next to the effects of auxin, which I introduced in my earlier video. So just to summarize the role of auxin, you find auxin is necessary for growth of plant, for the maturing of the plant, for the flowering process, as well as fruit development. Now, I did mention in my previous video that usually in questions, when it comes to flowering and fruit development, the focus will be more on the hormone ethane. However, I will briefly mention the role of auxin in relation to flowering and fruit development. Let's start with the first effect, that is growth. So there are two stages involved in growth. One is cell division by mitosis to increase the number of cells in the plant. And the second is cell elongation to help the plant grow taller. Then under maturing, we have three factors. That is phototropism, growth of the plant towards the sun. Then we have geotropism, that is growth of root downwards towards gravity. And the third thing is called apical dominance. That is when one shoot grows taller than all the other shoots. So for example, here we see there are three shoots that are like same length. So this is not apical dominance. But here you see one shoot growing very tall. This is apical dominance. So all this is controlled by auxin. Then we see the third effect related to flowering. Now auxin is not directly connected to the process of flowering. However, auxin prevents the flowers from falling off the stem. That is, it inhibits a process called abscission. Now the actual process of abscission will be discussed when we talk about the hormone abscisic acid. So just remember, auxin will help to keep the flower bud, the leaf intact or attached to the stem. And finally, for fruit development, we find auxin is involved in a process called parthenocarpy, that is production of seedless fruits. So I'll go into each one step by step. Have a look at the first example of growth, that is cell division. The role of auxin is very obvious in the process of cell division during tissue culture. Tissue culture is when we take a piece of tissue from a plant and then we grow it in a culture medium so that it becomes a clone. For the cells to divide, we need to add auxin plus another hormone, that is cytokinin. You see, auxin alone is unable to make the cells divide. Auxin must be present together with cytokinin, only then the cells will divide. So a relationship where both hormones depend on each other is known as synergistic. So in the situation of tissue culture, auxin and cytokinin are synergistic. Okay, but you must remember, sometimes in certain plant processes, auxin and cytokinin are not synergistic. they work opposite to one another, that we call as antagonistic. I will explain that when we discuss cytokinin. Now coming back to the role of auxin and cytokinin in tissue culture. Now, for a scientist to have the tissues growing into a clone, they must find a balance between the concentration of auxin and cytokinin. If you see here, you put small concentrations of auxin and cytokinin, but the same amount, the tissue will remain what we call as a callus. It will not develop roots, it will not develop a shoot. So what scientists tried second round was they increased the amount of auxin. Okay, so the auxin concentration was high, but the cytokinin concentration was low. And what they found is the roots started to form, but there were no leaves. So they had to modify the concentrations once again. That is, this time they reduced the amount of auxin and they increased the amount of cytokinin. Guess what happened? This time the leaves grew, but there was no roots. So you see, being a scientist is not easy because they have to keep experimenting. So finally, in conclusion, what scientists found out was that if the concentration of auxin and cytokinin are equal, that is either they are both equally low concentration or they are both equally high concentration, the tissue is unable to do proper cell division and will remain as a callus. But if we increase the amount of auxin to higher concentration and cytokinin remains in lower concentration, the callus can start to do cell division and differentiate to form roots. But there is no shoot. And if we reverse it, whereby this time we increase the concentration of cytokinin but reduce the concentration of auxin, what happens is the callus will divide to form a shoot, but there is no root. So the conclusion that scientists finally made was that the auxin and cytokinin do have to be in the same concentration, but they cannot be too low concentration or too high a concentration. There must be a proper balance. And then you get a clone with shoot and roots. So this is an example of how auxin and cytokinin are synergistic. This is the first effect. We move on to the second effect that is called cell elongation. So we know for a plant to grow, the cells will become longer. This is based on what is called the acid growth hypothesis, which in our syllabus we do not need to know the details of. So here I just explain very briefly about the acid growth hypothesis. Now let's say these three triangles here are auxin. So when the cell wants to elongate, auxin will diffuse to the cell wall of the cell and cause the enzymes there are specific enzymes that will help to break down the cellulose cell wall, making the cellulose cell wall a bit more elastic. So when water diffuses in by osmosis, small vesicles will form inside the plant cell, and these small vesicles will eventually fuse together to form a large central vacuole. Now, as water keeps diffusing in, turgor pressure will start to push onto the cell wall. In a normal situation, the cell wall will be hard, so the size of the cell will not be affected. But in the presence of auxin, the cell walls would have become more elastic. So what will happen is the turgor pressure will actually make the cells elongate. This is the acid growth hypothesis. Now remember, you do not have to memorize the details of this hypothesis. It is enough if you understand what this hypothesis is to answer objective questions. Let's move on to the second group of effects, that is the maturing of the plant. Okay. Now, the first one is phototropism, that is the plant's attraction towards sunlight. You see, if the sun is directly above, the plant will grow straight up. But if the sun happens to be to the left or to the right of the plant, it will cause the plant, not like in my cartoon, to walk to the sun, but the plant will start to bend towards the sunlight. Next, we look at the second stage of growth and development in a plant, that is the maturing stage. Firstly, we look at phototropism. Now, plants synthesize auxin at the apical meristem, and the auxin will diffuse down to the zone of cell elongation to elongate all the cells here so that the plant can grow taller straight up. Phototropism is when there is sunlight as the stimulus. So if the sun is to the left or to the right of the plant, what happens is auxin shows its specific characteristic, that is, it is sensitive to light. So now this part of the stem here is exposed to sunlight. So you see auxin will start to diffuse to the side that is not exposed to the sunlight. So this part now has a high concentration of auxin. And this brings to auxin's second characteristic. The first characteristic is that it was sensitive to light. Second characteristic is that it has an ability to promote cell elongation at the stem when in high concentration. So that means this part of the stem, the cells are going to get longer, whereas this part that has very little auxin is not going to be so long. So as the stem elongates, it will have to bend towards the sun. So this is what we call as phototropism. Let's talk about geotropism. Geotropism is related to the root. Phototropism was about the shoot. Geotropism is about the root. So the root also has an apical meristem where auxin is synthesized. Okay, now because of gravity, the auxin will sort of like collect or accumulate at the base of the root. Now here is the third characteristic of auxin, that is high concentration of auxin will inhibit cell elongation at the root. Just now when I talked to you about the shoot, remember high concentration of auxin will stimulate or promote cell elongation at the shoot, but it is the opposite at the root. So what's going to happen? The cells on this side are not going to elongate, but the cells that have less auxin above here will elongate. So this part of the root will get longer. So the length will cause the root to now bend downwards. So this is called geotropism. These characteristics can be a bit confusing. So let me summarize auxin in this graph. The y-axis shows growth response when applied auxin. Okay, and the x-axis is showing us concentration of auxin. Now we take the 10 to the power negative 12 to say about 10 to the power negative 9 as low auxin concentration, and the concentrations above that as high auxin concentration. Then we see the y-axis where they show 100% increase, 200% increase in the growth of the plant. This we consider as stimulating the growth of the plant. Whereas over here you see it's going negative 100. That means the plant ends up going shorter. So this portion we say the plant is inhibited. Now the graph here has got three organs, that is the roots, the flower bud and the stem. But for our syllabus, the focus is only on the root and the stem. So I'm going to completely ignore the bud. So if you observe, the graph for the root shows us that low concentration of auxin actually stimulates elongation of root up to maybe say about 10 to the power negative 9 molar concentration of auxin. After that point, the concentration of auxin is considered high, and you see the root is actually getting shorter. So high concentration of auxin inhibits at the root. Now if we compare with the stem, at the stem you see low concentration of auxin, there is no growth. So actually low concentration of auxin has no effect on the stem. We cannot say it inhibits, it has no effect. Okay, but when high concentrations of auxin are used, we can see there is a stimulating effect on the growth of the stem. But very high concentration of auxin actually has an inhibiting effect to the growth of the stem. So this summarizes the characteristics of auxin in relation to the plant maturing. We look now at the third aspect of maturing, that is something called apical dominance. Now if you look at these plants, all the plants down here are almost of the same height, except for this one tall stem. So this stem shows us that it has a lot of auxin and started to grow very fast, showing apical dominance. Dominance means like powerful. So that is why you see, if you are looking after your garden, when you have stems that are too tall, we like to cut them off. Okay, I'll explain why we do that. That process is called pruning, P-R-U-N-I-N-G. Why do we do pruning? Is because you see like in this first picture here, okay, there's one single stem going up. So this single stem is going to cause apical dominance. But if we cut off that single stem, you see out of nowhere, originally here you can't see anything. But the moment we cut off the main stem, from the side here you get two new stems. This we call as the lateral shoots. So in gardening, like I said, when we do pruning, when we cut off the main stem, we end up getting many small branches. So the plant will look thicker. Right? So this is caused by auxin. Moving on, we look at the role of auxin in flowering. Okay, for the flower to bloom, that is actually the job of ethane. But what auxin can do for flowering is it will produce the flower bud, and then it will make sure that the flower does not fall off from the stem. Okay, so auxin also makes sure leaves don't fall off the stem. So this is what we call as inhibit abscission. The process of abscission, as I mentioned earlier, I will discuss when we reach abscisic acid. The final role of auxin, that is in fruit development through a process known as parthenocarpy. Parthenocarpy is the production of seedless fruits. Okay, I'm sure eating watermelon like this is something very common for you. But when I was growing up long ago, these are the types of watermelon that I eat. Watermelons that have seed. This is the natural watermelon. Okay, now why is it watermelon became this? Okay, seedless form. Actually, it's just human laziness. Okay, humans for some reason decided they don't want to eat watermelon with seeds. So they found a method where they take the watermelon flowers, whenever the watermelon plant flowers, they spray it with auxin. So what this auxin does is it will kill off the flower, but it will stimulate the ovary in the flower, okay, to become a fruit. So since no fertilization happened, there is no seed. So this is the role of auxin. So next, I will see you for cytokinin. All of you have a nice day. Bye-bye.